Hello everyone, and welcome to our first overview lecture here. This is a complete summary of Chapter 18, so yes, all you have to do is follow along with your original PowerPoint, and that's all I'm going to do here, but I'm not going to be spending too much time or slowing down at points, because at this point, the expectation is like the hypothalamus is right there, you are able to identify that this is the thyroid gland, this is the thymus, that's the expectation at this point, is that you already know a lot of these overview things. So if you're coming in thinking, hey, I'm just going to listen to the overview lecture and that is it, I don't think you're going to do quite well. This lecture is designed for students that have gone through all the material and kind of want to confirm that they're understanding the main big picture concepts. As usual in class, I stress 4 key points that you need to get to memory. Now, granted, you can try to get these to memory, but they're not usually going to make sense without a lot of background knowledge, which is why I continue to stress you should really listen to all the lectures first and then follow along here. Or if you are listening to this ahead of time, this might not be too bad to give you an overview and then go through the detailed lecture itself. Either approach would be fine, but don't think just listening to this overview lecture is going to help you do well in this entire course. It's going to take a lot more than just that. Alright, but we're going to start off and we're just going to go through kind of the major points here, and I'm not going to be stopping at these objectives and those sort of things. Obviously, as I've stressed, you should be able to answer all of these. Like, if you went through this and can easily describe these objectives, you probably know the content, and it is kind of that simple. If you can't, if you struggle at any one of these, that means that's where you need to spend some of your time and focus on that area. Alright, but let's get into it. Obviously, this was all about the main definitions related to kind of endocrine system and then talking about these various forms of communication that we see on the side here, right? So, hands down, you should know what the endocrine system is. You should know it's long-term process regulation, and it uses the chemicals hormones. That's the major differences. It's regulating our long-term. The best thing to think about is regulating blood glucose, regulating blood pressure. Those are the easiest things that I think are able to relate to the endocrine system. If you're like, yeah, that makes sense, that's where you should be. You know that these hormones bind to receptors. At this point, you should be thinking intracellular or extracellular and being able to relate the extracellular receptors to peptide hormones and the intracellular to the steroid derivative. Or we can also, instead of saying peptide hormones, we can say water-soluble and then steroid derivative or lipid-soluble hormones, right? Intracellular, extracellular receptors. So notice, when I look at these terms, I'm able to expand almost immediately. All of these concepts should make sense. Then we move on to this. As I said, this is pretty much definitions and should be really basic. You should know and relate it to, like, direct communication is the heart. Those intercalated discs are literally going to allow calcium to move through the heart and coordinate the heart contraction, which we'll be talking about in the second unit coming up. All right, paracrine, you should associate para being within a tissue, associate with healing process, autocrine. We're at this point, you should be probably through chapter 22 as well, because that's part of the first unit in my class. So you should know helper T cell activation was really the only kind of time we've really talked autocrine communication. That was, of course, when it binds to a B cell, right? Binds to the B cell, and when it binds to that B cell, or just an antigen presenting cell in general, it was going to stimulate and activate itself. All right. Here's endocrine. Again, we use the bloodstream in most cases and the use of hormones. It's really the bloodstream that defines the endocrine, where here, synaptic communication, we're still using chemical signaling. Don't forget that. But of course, those are neurotransmitters. So neurotransmitters in the case of synaptic, endocrine, you need to be hands down thinking hormones within the bloodstream, here within the synaptic cleft. Again, these are all of the basics that you should have down already. I'm not going to spend too much time here. Obviously, endocrine strength is based on how much of a hormone is secreted, and then, of course, most of that's regulated by negative feedback, which should all make sense at this point. Here, we know this is neural signal, how frequent the neural signal gets sent. The faster the signal, obviously, means the stronger the signal or the stronger the response. All right, so when we got deeper into hormones, again, this was really telling you what hormones can do, but by this point, you should already know there's amino acid derivative, there's peptide hormones, there are lipid derivatives, and again, we stress the point with lipid derivatives that they were able to pass the cell membrane quickly. We stress that peptide hormones, those are going to be amino acids. Those are not going to pass the membrane, so those are going to be binding all to the extracellular. And then with amino acid derivative, there was only one key lipid-soluble hormone, and that, of course, was the tyrosine derivative thyroid hormone, right? all the other hormones are technically going to be water soluble. The only exception was thyroid hormone. So we got to this part at the end, so when you read this, that should now make sense when we talked about how lipid-soluble hormones bind to intracellular receptors, and when they do, what did they do? They basically activated a gene. They almost act as like co-activators. And then, of course, when we talked about peptide hormones, we talked about how they bind to the outer, or again, water-soluble hormones, they bind to the extracellular receptors, and that led to secondary messengers, and we'll get to that later, right? And so we're not going to spend too much time here. At this point, do you know thyroid is a lipid soluble? Do you know it's tyrosine derivative? Do you know norepinephrine and epinephrine are the catecholamines? At this point, if you're familiar with that, I would say you have hands down the main points. And then know your tryptophan derivatives are melatonin and serotonin. I think that's kind of the big focal areas. But, you know, the kind of A to B students, it's knowing that, you know, dopamine is a reward response. It's part of a catecholamine, and then knowing the minor effects of these. But melatonin I tend to test on a little bit more because you should associate with tryptophan and sleep. That tends to be my major one. I would argue norepinephrine and epinephrine and melatonin, along with thyroid hormones, should be the three on this slide, or four if you consider norepinephrine and epinephrine as a separate one. But those are the major ones you should hands down know. All right, again, these are kind of key terms that you need to know. You hopefully understood the chemical cascade concept, and that should now make a lot more sense that you have experience with the secondary messengers that we talked about, and we talked about kinases, right? A kinase activates an enzyme, and that can activate, or actually a kinase can activate another kinase, which can activate another kinase, and all we're talking about is a chemical cascade that essentially leads to a cellular response. The main point is cells can basically regulate the chemical cascade, and that's how cells regulate their response. All right, but again, at this point, this is all kind of tying concepts together and make everything seem easier if you were linking all these concepts, right? Now here, these concepts should be well understood. Obviously, we're focused on the lipid derivative hormones. You should definitely know they're a derivative of cholesterol. That is one of the key things that I hands down stress, just like if I say peptide derivative, you should know they're derived from amino acids. And if I say amino acid derivative, you should know it's either tyrosine or tryptophan, right? And there is a second class of lipid messengers, and that second class of lipid messengers was the eicosanoids. I don't touch too much on them. The major point of eicosanoids, you should associate them as paracrine signaling, and obviously since we've had a concept and we've had terms, paracrine signaling previously, you should be able to associate and link those. Those were a concept linked off of this. And the main one I focus on is the prostaglandins, because these are what are going to stimulate the essentially vasoconstrictors to constrict when there's damage to blood vessels, but kind of helps regulate and coordinate some of the clotting that has to go on. All right, but that's the extent I want you to know for now. Obviously, this next slide just continues on the major points related to your steroid hormones. Again, just reviewing this, because this is a review lecture, you should instantly be able to name off these hormones. What are the sex hormones? Well, testosterone, progesterone, and estrogen. What are the hormones of the adrenal cortex? Well, aldosterone, cortisone, and androgens, right? And this was zona reticularis, which you should know right away. This was zona fasciculata, and then this was zona glomerulosa. Like, these are all things that should make sense just from seeing these slides. And if you're like, yeah, this does, that means you've connected all the major concepts, because going through a review of my PowerPoint should be really basic at this point. All right, and then the kidneys, of course, secreting calcitriol. Do you know what this does? Well, it's a derivative of vitamin D that helps stimulate the absorption of calcium at the intestines, which is how it differs from parathyroid hormone. But it does, like parathyroid, it does stimulate the osteoclast to break down bone and release calcium into the blood. So it helps increase blood calcium levels. Notice, when you're going through my PowerPoints, are you able to do this? If you are, you're working at a really great pace and understanding the major points. Like I've said, I would encourage you, if you haven't read this, I would just read over this. It's not going to give you too many extra details. It might help solidify some interesting concepts, but I didn't cover it in too many details. But notice, coordinating local cellular activity, obviously paracrine signaling. It's telling you what their kind of minor effects are. All right, but we spent a lot of time on this. Again, you need to know the major points. When it comes to water soluble, major point is cannot pass the bilayer, which means it has to bind to the extracellular receptors. That's going to be all of the peptide hormones, and it's going to be most of the amino derivatives except for thyroid hormone. All right, These are considered free hormones because they cannot pass the bilayer, so they don't need transport proteins. But what in effect that means is it means enzymes can attach to these easier and break them down. That's why these are shorter-lived molecules in comparison to your lipid-soluble. Major points of lipid-soluble, these are nonpolar hormones that are able to bypass the bilayer because they're literally lipids, and it's a lipid bilayer. And so they bind to intracellular receptors. All right, and those are inside the cell. These are all the steroid derivative hormones and thyroid hormone that is considered lipid-soluble. And these have to be bound hormones. They have to be attached to transport proteins. And you'll learn about transport proteins come chapter 19, which is whole blood. But those are some of the main proteins in our blood, is literally to transport steroid-based or lipid-based molecules. Because, again, they're lipid-soluble, which means they would be passing through every membrane. So they need to be bound to something like, and that's basically serves as almost like a bus stop. All right, so that was quite a first part just to get through. And so now we move into the second part, and here it was a general overview of the endocrine organs. So really this is going to be some of the more basic sort of concepts. So when I introduced the hypothalamus, you should be able to get in greater details than this, but the major point was it's the master endocrine gland or the president of the endocrine system, right? That's what I was really stressing with it. And you should just understand there's a lot of sensory input that goes to that organ, and that's how it is really heavily controlling our body. Now, I don't get into that great of details of the specifics, but the major point is you should know this is the main overview gland that controls everything. Most sensory input is going in there. A lot of the unconscious things and a lot of the details that we did not get into are ultimately regulated at the hypothalamus, but we're going to focus on very specifics to give you a good general input. Then we moved on, of course, from the hypothalamus to the pituitary gland. There we talked about, 9 different hormones being secreted, 7 of which are produced at the anterior pituitary, and remember, 2 were just stored in the posterior pituitary, but those were produced by, again, neurons that are stored in the hypothalamus. Their cell bodies are found in the hypothalamus, but their axons lead to the posterior pituitary, right? And so we're just building off concepts. Thyroid, the major point with the thyroid gland is it secretes T3 and T4 that supports normal metabolism and is important for development in general. Again, these are the big picture points. Remember, we also talked about heat regulation, and we did get into hypo versus hypersecretion. So that's where we're heading with that. And if you're thinking about those concepts and like, yeah, I remember those, again, you're doing really well. The adrenal medulla, we said emergency metabolism, metabolic activity. That's going to be via adrenaline. So epinephrine, norepinephrine are released here, and that's during sympathetic stimulation to really give us that boost in our heart rate, boost in breathing rate, really circulating the oxygen to get us going. So that's the point of really epinephrine, norepinephrine is the emergency sympathetic response. Hence, think of that hormone as epinephrine, norepinephrine as the emergency metabolic hormone. The adrenal cortex, we really talk aldosterone, which if you remember was salt balance. You should be thinking sodium and potassium exchange, right? Sodium in, potassium out. And similarly, it's the stress metabolic response there. We're talking cortisol and cortisone, which, again, our body goes through some normal daily stresses, which is fine for cortisone to be secreted, like waking up is a stressful time for our body. Day-to-day stress, we get these little shots of adrenaline, and then when we're dealing with stress for a longer time, cortisone and cortisol help with that a bit, but again, you don't want that too much. You want to keep those levels low, and that's usually kind of hard to do when we live in a very busy environment. Pancreas, we said blood glucose regulation. That's the main point. Again, you got alpha and beta cells, you got insulin and glucagon. You really need to break those details down, but it was really beta secretes, the beta cells secrete insulin that helps reduce blood glucose levels, typically after meals. Now we say before and during and after, but it's really when blood glucose levels rise. So it's really going to be secreted heavily after a meal. Probably, I would say, during and after. All right, and then glucagon, of course, was secreted by alpha cells to help regulate blood glucose when we're in kind of fasting mode, and so that'll keep our blood glucose normal when we don't have access to food for 4, 5, 6 hours, basically between meals. All right, pineal gland, that's the sleep cycle regulation that secretes melatonin. That's what you really need to know. Parathyroid, blood calcium regulation. It helps increase the amount of calcium in the bloodstream. It does so by stimulating the osteoclast, and it does so by stimulating the reabsorption of calcium at the kidneys. Heart is going to secrete BNP or AMP. Those are also called natriuretic peptides, which we get to later in the class. But again, it's the only hormone that reduces blood pressure, and I guarantee you I will be asking that several times. Which of the following hormones reduces blood pressure? And then when we got to the kidneys, again, producing red blood cells, calcium regulation via calcitriol. Those are kind of the main points there. All right, so that was the overview. You should have that down. A lot of that should have just seemed pretty simple at this point. But then we got into the pancreas, and like I've said, anatomical descriptions, knowing endocrine versus exocrine, which obviously at this point, you hands down need to know the pancreas is both. It's endocrine and exocrine. And when it's exocrine, we call it the pancreatic acini cells. And the pancreatic acinar regions are the regions where it's dark pink. And then when we get into the endocrine regions, which are the pancreatic islets, that was going to be these lighter islands, and they're just small islands. And like I've said, this probably isn't the best image of it, but main function of the pancreas, obviously when it comes to digestion, and that's not what we're focused on, but it is to aid digestion via enzymes. That's one of the major functions from a digestive perspective. But number two, and since we're talking endocrine, regulate blood glucose levels using insulin and glucagon. So just think that's the big picture function of the pancreas when it comes to the endocrine system. All right, insulin and glucagon, like I said, with insulin, the major point that you should know is it's a peptide hormone. Obviously, it's not the 7 that we talk about that are lipid soluble, but obviously what you need to know is it's secreted by the beta cells. All right, so insulin gets secreted by beta cells in the pancreatic islets, and those beta cells function to reduce blood glucose levels, or we can say insulin functions to reduce blood glucose levels. Again, secreted primarily during and shortly after meals. Technically secreted before meals as well. Like the sight of food, it starts to rev up, but really it's going to be primarily during and shortly after meals. All right, so hands down, you need to know that what it does is it targets the liver and skeletal muscles to increase the glucose uptake. So by causing glucose uptake in our liver and muscle cells, that reduces the blood glucose. So understand that concept. So insulin really is stimulating our cells after we consume a meal to absorb nutrients. That's the easiest way to think of insulin. We focus on blood glucose because, of course, glucose is going to be one of the major substances that we get in our diet, along with fats and along with amino acids, right? So notice increases glucose and amino acid uptake, utilization, and helps with ATP production. So just think, insulin after meals to help our cells uptake those nutrients and utilize them, and then continue with utilization just to maintain our body, because of course we have metabolic activity, we constantly are regulating energy. Now notice, once it uptakes those, then it stimulates those cells to help form glycogen, which is the long-term storage of glucose, helps form proteins via amino acids, and helps form those triglycerides, which are essentially the lipids. But hands down, if you have these main points and are connecting all those, that's the breakdown. Similarly, you should just do the opposite for glucagon. If you get really good with insulin, just think glucagon is the exact opposite. You have your alpha cells, they secrete glucagon. The goal is to increase blood glucose levels by still targeting the liver and skeletal muscle, but instead of building up nutrients, we're breaking down those storage nutrients. So breaking down glycogen, which is the storage form of glucose, breaking down the triglycerides into glycerol and into fatty acid chains to be used in the bloodstream. Major points right there. You need to know, hands down, this pancreas. Now here, this is more on the lab side of things, but obviously you're going to see these on quizzes. If you're watching this after you've taken a quiz, you've probably already seen a couple of these images. But right off the bat, I know that these are the pancreatic islets. I can easily follow up with and say, name what's secreted from here, which is insulin and glucagon. Similarly, I can say what are these dark pink regions over here? That's the pancreatic acinar regions, and you would need to know what's secreted from there is digestive enzymes. That's all you would need to be able to do. Again, overall function of this would be, since it's the pancreas, it regulates blood glucose. And if I just say, what is this histological slide of? It's the pancreas. So kind of a lot of things covered here. I didn't go through it in kind of the logical order. Obviously, I should have started with what is this a slide image of being the pancreas? What are these? But regardless, you would have gotten that from the lab lectures. Again, overview lecture, I go very, very fast, as you can see. All right, and then that brought us to diabetes. And really the main point that I would stress that you focus on is knowing that type 1 is insulin dependent and type 2 is insulin resistance. Those terms at this point should give away what this means. Type 1 being insulin dependent means hey, the person relies on insulin injections because their beta cells are faulty, or typically it can be an autoimmune disease, which means their beta cells have been attacked. All right, so it's rare. It's more of an genetic case, but it's on the rarer side. Where type 2, since it's insulin resistance, this is where basically your body, these receptors have been downregulated on normal insulin production, isn't stimulating the cells like it needs to be. Often associated with diet and often treated with medications, but exercise and dieting can also help tremendously with it. And obviously, if you want to know more and get into more details, I would recommend reviewing Figure 18.18. The book does an excellent job discussing all the kind of issues associated with having too much glucose in your blood, which, if you just think about it, think about sugary water and how much stickier that gets. So if your blood glucose isn't regulated and there's high blood glucose, obviously that leads to some complications, right? All right, so that gets us through the first section of this. But what I would encourage you to do, can you answer these questions? If you can, you're probably well prepared. All right, so then the second lecture, and this is probably what I'll get through for the review today, and then I'll do the second review in a second part. So I'm going to break this review into two parts because we're already going on about 20 minutes here. That means the second part's probably going to take about 20 minutes as well. All right, but when we went through this, we really got into endocrine control, and the main point, there was 3 control units, and at this point, you should be able to list them. There was hormonal control, right? So when one hormone binding to its target caused the release of that hormone. So thyroid stimulating hormone binding to the thyroid gland, causing the thyroid gland to release and produce more thyroid hormone, right? That's hormonal control. Then we had humoral. Humoral was when we sense changes in the bloodstream. So in that case, humoral control, really the best examples are like when our blood glucose rises, insulin gets secreted, right? Blood glucose change, so insulin needed to be secreted. When we're thirsty, our blood pressure is lower. So because of that, the salt concentrations are changing. The body senses it, and it causes the release of ADH. Notice that's humoral changes responding to changes in our bloodstream. And then the final one, of course, was direct control via neurons, and so sympathetic control. And really the sympathetic control here, they're only showing this one, but you can also put, if you remember, we talked about renin. Renin releases sympathetic stimulation as well. Okay, and that's something we're going to talk about later, but you should know these 3, and you should know this is mostly negative feedback, especially because we did that activity in class, that worksheet that I handed out that was quite confusing, but went over the feedback loops, right? So early on, when we talked about the hypothalamus, we said it was the master endocrine gland. You really needed to understand its 3 control methodologies. And so it controlled the anterior pituitary. Our focus was on the release, or the use of releasing hormones to cause those 7 hormones of the anterior pituitary to get released into the bloodstream. So remember, anterior pituitary regulates the pituitary gland through regulating hormones, but the ones we concerned ourselves with was releasing hormones. We'll get to those, but you should know the main ones. And then with the posterior pituitary, we saw that here's the neurons, right? And those various neurons, we produce either ADH or oxytocin, but the reason why they're not considered, why they're not neurotransmitters is because they're going to release the actual molecule, the chemical that's stored there in the axon terminal into the blood, which is why it's still technically a hormone. All right, and then the adrenal medulla was sympathetic stimulation that caused the release of epinephrine, norepinephrine. But again, various forms of sympathetic stimulation will also lead to the release of renin. All right. Now, hormones, again, releasing hormones, they target the anterior pituitary. This is what we've been discussing. Really, we've gone through these over and over again. Hands down, I've stressed ADH. You definitely need to know that one. Oxytocin, I'm telling you, it's going to be on the exam, so know what it does. Oxytocin targets uterus, causes contractions. Oxytocin targets the breast for milk release. Remember, prolactin targets the breast for milk production. So I told you that those were going to be on the exam. You better be prepared to see those. All right, ADH is a big one. It's a must know. And then just understanding this more conceptually is really key there. That's how the hypothalamus regulates the anterior pituitary. It does so by causing releasing hormones to target the anterior pituitary. All right, and so these were the releasing hormones that I said you needed to know. Again, these are produced by the hypothalamus. and they target the anterior pituitary. So thyrotropin releasing hormone, or TRH, and you can see that right here, targets TSH, or leads to the release of TSH by the anterior lobe. CRH, corticotropic releasing hormone, targets the anterior pituitary to release ACTH, and ACTH will of course target the adrenal gland. So we're seeing multiple cascades of one hormone controlling the release of another, and then this hormone controlling the release of another. That's all hormonal stimuli so far. None of this so far is humoral. We're talking all hormonal stimuli. I hope you made that connection when you were reviewing notes, that everything we're talking about right now is hormonal stimuli, but if not, hopefully the review gets you to catch it and gives you a little bit more of that, like, oh yeah, I should have known that. Now this is making even more sense. All right, and then, of course, gonadotropin releasing hormone, that's going to cause the release of FSH and LH. That one's a little bit more challenging because it does cause the release of two hormones. All right, but these were the ones to note. These are releasing hormones produced by the hypothalamus and target the anterior pituitary. All right, so that kind of summarized the hypothalamus. Really wasn't histology that I test on, but then we move on to the pituitary gland, and we talk about releasing 9 peptide, but again, only producing 7, and those 7 are produced here. The other 2 that are stored here are produced by the hypothalamus neurons. I've stressed that over and over. Again, with them being peptide hormones, you should know they bind to extracellular receptors. So notice you're combining a lot of concepts, and reviewing this PowerPoint and seeing all these concepts that you should be able to connect because you've seen the future content should really be tying all these things together. All right, the infundibulum, just remember I tend to label that up on the lab exam, especially I use usually a model image, and I just label that region, and so know it's the infundibulum. All right, but 2 distinct lobes, you have the anterior and the posterior pituitary. Remember, when it comes to histology, there's 3 histological layers, and you need to know them as histological layers. Here, I will accept both histological use terms or anterior pituitary, but on histology, I will only accept the term pars distalis. So on like a model, if you said pars distalis here, I'd give you credit, and I would give you credit for anterior pituitary. But when we go into and we start looking at histology, so let me just hop ahead here. So here, you must use, this is pars nervosa, pars distalis, but I'll get to that in just a little bit. All right, but make sure you know these. No pars nervosa, as I'm hinting at. All right, and then you really should know these general functions. I know there's 7 hormones, which just makes it seem like it's very hard to narrow them down, but really it's TSH stimulates the release of thyroid hormone by targeting the thyroid gland. ACTH targets the zona fasciculata, stimulating the release of cortisone, corticosteroid, and cortisol, i.e. under stress. So notice that's kind of like sympathetic stimulation almost, but basically stress is causing this release. All right, FSH, LH, like I said, FSH, no for now. Just know it stimulates the egg development and sperm development. And then with luteinizing hormone, just know that it stimulates the release of ovulation right here. That's really all I care about. Prolactin, producing milk, I've said, and growth hormones stimulates growth. And don't forget, associate growth hormone with somatomedins. All right. There's your ADH, there's your oxytocin. The only thing you needed to add on to it, obviously you needed to know this. These are the details that get the A students the A and the B students to just miss the A. It's really knowing that the supraoptic nuclei is the one that produces the ADH, all right, and then the paraventricular is what produces oxytocin, all right, and you just kind of need to memorize that. I don't really have any great way of knowing that. Just it's remembering paraventricular is oxytocin, supraoptic ADH. All right, and then don't forget the inferior hypophyseal portal system is what carries these hormones into circulation. All right, and then we reviewed receptors, and I'm not going to go into receptors because I've already reviewed that in some of the concepts already, so I'm just going to keep on moving. You should have this concept down, extracellular, intracellular. We're definitely going to retouch on this when we get to, of course, the last lecture, right? All right, so we finished off this with really the hypophyseal portal system, which you should know is what links the hypothalamus to the pituitary gland. That's the big picture sort of definition, but if I'm going to give you a more specific question, what you need to know is that we're talking about the superior hypophyseal portal system is what ties the hypothalamus and carries your regulating hormones to the anterior pituitary, while the inferior is what carries hormones of the posterior pituitary into circulation. So very different. Regulatory hormones travel using the superior hypophyseal portal system. Now note, you should know that these are fenestrated capillaries. This is one of the details that overlaps with chapter 21, I believe, when we get into blood vessels. So for now, know that these are capillaries that have pores in them, and capillaries are where exchange happens. And so obviously if you have pores, that increases the ability of that vessel to exchange, where some don't allow for this big of exchange, basically the basic nutrients. Well, the hypophyseal portal system is for larger things like peptide hormones. All right, and then again, histology. Make sure you know these layers. Don't think just because we have two examples where it's on the left that pars nervosa is always on the left. Notice on here, this is the lighter side. So conceptually, when it comes to histology, the lighter side is pars nervosa, which means oxytocin and ADH are released from there. The kind of this area right there where we see this lighter color of pink compared to here where it kind of darkens up, that's a great example of your pars intermedia. So that would be fair game for me just to label. I don't usually follow up any questions there. And then you would need to know that's pars distalis. I can ask what portal system links to this. That you would have to say the superior hypophyseal portal system. What system carries these hormones that are released into circulation? That would be the inferior. So I can certainly expand on some of these questions. Usually those would be really detailed, so those would be more on the extra credit side of them. All right, but pars distalis, you need to be able to list at least 4 hormones being secreted from them. It's 4 of 6, and to be honest, I don't care if you know melanocyte stimulating hormone is technically secreted from pars intermedia. I don't usually get too carried away. I just want you to be able to list 4 of the 7 hormones. All right, so know your histology. And then the remaining things here, we're just showing you the regulation side. And at this point, this should make a ton of sense now. It might have been really confusing when I first introduced you to this, but you should be able to go back to this and say, yes, a lot of these concepts make complete sense to me here. If they don't, well, which part doesn't? Like, this is showing you, hey, this is hormonal stimuli to finally releasing the final hormone. And of course, again, you should be able to say, hey, thyroid hormones are going to stimulate and regulate metabolism, right? Oh, glucocorticoids, that's the stress hormone that causes our body to produce glucose at the expense of muscle tissue and at the expense of fats. All right, so this should make sense here. And then similarly, a figure like this should now make sense. So you should be able to say, hey, ACTH, I know that was secreted by the pituitary gland. And notice, these are all the hormones secreted by the anterior pituitary lobe. This was the pars intermedia. So this is pars distalis, pars intermedia, MSH. And then notice ADH and oxytocin. Oxytocin is the paraventricular nuclei. And then that was the supraoptic. Okay, and so notice they're showing you this sensory side of things that, hey, there's sensory going on, and that would be humoral to cause these releases. This is more on the humoral side of things. Although I would assume that would actually be stretch, so that's a little bit different. But sensory stimulation here that they're talking about is stretch. Here, osmo is more on the humoral side. All right, and then don't forget, obviously, we can have what happens when hormones are secreted at various levels that they shouldn't be, but this, of course, we're talking about growth hormone. These are basic definitions, and usually students don't struggle with these concepts when I test on them, so I'm not too concerned there. But the big thing to note is growth hormone leads to the production of somatomedins, and again, you can see that and backtrack it here. Growth hormone to the liver, liver converts it to somatomedins. Somatomedins essentially act as something called insulin-like growth factors, ILGFs. Not that you need to know that, but basically it stimulates growth. That's really all it does. So notice insulin-like growth factors. All right, and then acromegalia was when high levels are continuing. That's basically getting your bones bigger, wider, not longer. All right, but that takes us to the second lecture, and I'm going to end the review there. That way it's not too huge of a review. It's at 35 minutes. I kind of expected this to be one of the longer reviews. Usually it's going to come in around 100. It's going to come in at right around an hour and 10 to an hour and 20 minutes. So me cutting this here is kind of a good point. Hopefully when you're going through this, you were processing most of it. If you weren't, I would slow down and go through some of these and work out some of these kinks. If you went through this and felt really confident, then watch the second review. But very similar, the second review is going to move at this kind of fast pace. Again, if you're like nodding your head as I'm describing this, saying, yeah, that all makes sense, or hopefully catching a few of those, oh, I didn't realize that, but now that makes sense. That's the point of these. If this was all confusing to you, you have a lot of work to go back and do, and make sure you listen to the lectures in full. Look out for the second review lecture for the endocrine system. Take care, everyone.